Hello everyone, this is Mrs. Travai and this is chapter 3, Analyzing DNA Restriction Digests. And we're going to take a look at how restriction digests work and how we use them in this course. This slide should be pretty familiar to you by now. And at this point, we've done all these steps, but now we need to verify the size of our restriction digest and actually take a look at our MiniPrep to see if we were successful. So let's move on. And we're going to take a look at where the DNA was inserted. And if you remember, the DNA insert here, this is the piece of DNA from the Landoltia punctata that was spliced in and ligated into a vector. Our vector is a plasmid that has this SFI-A and SFI-B site. And it helps us to insert the DNA in a directional fashion because these sites have to match the SFI-A and SFI-B site on the insert, which means that the DNA needed to be cut with those enzymes and the vector has to be cut with the enzymes in order for them to be ligated together. Now, once this is done, this was inserted into or transformed into the bacteria, but where did this cDNA insert go in? It went in right here. Here's the SFI1A site. And if you notice, there's a different code for the SFI1B site. They're not the same. Again, this allows us to place the DNA into this section in a directional manner, which means that the DNA insert can't be flipped over and reversed when we're doing this. So let's take a look, go a little further, and take a look at our DNA MiniPrep. And we were pretty successful. Those groups that maybe weren't had to go back and redo their MiniPreps. But we picked a colony, we grew it. After doing PCR, you put it into solution 1. We froze it at that point. But what happens after it's suspended in solution 1? Basically, the cells denature, and by freezing it as well, the bacterial cells that are in there rupture and release all the contents of the cell into that solution. Once that's done, we then add, we rupture the cells here, and then in solution 2, we're denaturing the other lipids and proteins that are in there, but leaving the RNA and the DNA pretty much intact. What we then do is add solution 3 to actually bring this solution back to a stable pH, because we added a base, a strong base, and then we add a acid, acetic acid, and you could smell it when you're working on a MiniPrep. It smells like vinegar. And you put solution 3 in to be able to buffer it and put it back into the right pH. So now we're going to, we spin our supernate, we spin our tube, our MiniPrep tube, and this is a step where you pour it into the column, right? So you load the supernate into the column. We have our collection tube underneath, and we're going to spin it at the fastest speed on the centrifuge. And what will happen is you spin out the RNA and the proteins, and most of that is going to come through the column, and what you're left with in the matrix of the column is the DNA and some residual material which is not DNA. So we have parts of the cell that may get stuck in there that we need to still get out, which is the RNA and the proteins, some residual RNA and proteins that are left there. We then put wash buffer, which is an ethanol solution, and we add wash buffer and spin it out again. And what happens? The DNA precipitates onto this column because DNA is not soluble in ethanol. And it helps to precipitate the DNA in the column and get caught in that column. What'll happen is everything else, the bonds are broken in the matrix, and it allows those materials to wash through. So this is all debris that we don't need anymore. So we're going to dump this flow through and spin it again. And this is a step where we spin it twice, and we let the column air dry a little bit. And then once we're comfortable that we've air dried and spun out all the wash buffer, we then can add the elution buffer. And it's critical. This step right here is very critical, because if you have any ethanol left in your MiniPrep, it will not be able to sequence. Will not be able to sequence your sample, and you'll know when you load the sample into the gel, because your samples will float away. So it'll make it light and actually drag the samples upward. So right now, at this step, we add the elution buffer, and we elute the DNA. And again, this is a step where please don't throw this out. We actually spin them into yellow tubes, And you're going to cap the yellow tube, throw out the column, cap the yellow tube, and write MiniPrep on the top so that we know that's the MiniPrep. So that's been done already in lab. And right now, let's take a look at what's actually happening. What happens when we use restriction enzymes? So the next step would be to digest the DNA in a restriction enzyme to be able to analyze the size of the insert and to verify that we have DNA in our MiniPrep. So here's giving you an example. This is not the enzymes that we use, but here we have a EcoR1 site, a PST1 site, and a HindD3 site. Remember that these restriction enzymes come from bacteria. They're purified from bacteria, and we use them because bacteria naturally have the ability to cut DNA at specific restriction sites. So by doing this, if I digest my samples, so let's say I have these two plasmids, and I digest my samples with EcoR1 and HindD3, that's a double digest. So I put two enzymes in. It is only going to cut where the R1 sites are. So here's an R1 site. There's an R1 site on this plasmid. And the HindD3 is down here on this plasmid A, and again, it's on plasmid B. And again, if we purify the fragments and ligate them in, you could cut and paste these fragments together so that you could take a piece of DNA from another location or another organism or another plasmid and insert it into another plasmid so that you can actually create a new vector or a new sample which you'll use to transform. So this is what was done to actually create your samples. So how do restriction enzymes work? Basically, they are called endonucleases. They cleave DNA at a specific sequence. They protect, in nature, they protect the bacteria from phage infection because viruses, bacteriophage viruses, are out there infecting bacteria. And what happens once they bind to the surface of the bacteria and insert their genome, what will happen is the bacteria, if it has the right restriction enzymes, it can attack the bacteriophage and digest the phage DNA. So in this case, it's an EcoR1 bacteria, the first enzyme really found. And this cuts at specific points. So in this case, it shows you where the green arrows are coming down is the initial breaking of this backbone. And then the hydrogen bonds between the bases are broken to create what are called sticky ends. The reason these are referred to as sticky is because they want to make hydrogen bonds to the other bases or complementary bases. So it wants to come together again. So let's take a look at clicker question 1. Okay, open the quiz up in EDU 2.0 and answer your questions. Now, why don't the restriction enzymes degrade the cell's DNA, the bacterial cell's DNA? It's a good question. The bacterial DNA is actually protected with methyl groups, and the areas that the bacterial DNA, because it has those methyl groups, these endonucleases can't wrap themselves around the DNA and cleave at those points. So it methylates its own DNA to prevent this from happening. The bacteriophage, on the other hand, does not have methyl groups on its DNA, and it quickly digests. The endonucleases will quickly digest the DNA. Now, methylases would go ahead and actually methylate this DNA as well, but they have so much more endonucleases that it's much quicker, and the endonucleases get there first. So, clicker question number 2. And basically, like I said, we found that each organism has a specific set of restriction enzymes. And E. coli, they take the genus and species name and create a name, EcoR1. BAM, H1, PVU1, PVU2. And these are all sold by labs that purify these enzymes and provide them for sale. So you can pretty much go to any of these labs and look up how to purchase these samples. So this is what we've referred to as sequence recognition cleavage. And if you take a look, there are some enzymes that create 5' overhangs. And what that means is that the overhang here, if you take a look, is the actual breaking is creating what's called a 5' overhang. If it creates the opposite, then you create what's called a 3' overhang. Okay, and the last type of enzyme that we can see, and we don't necessarily use this in the lab for our purposes, but we can have one that's a blunt cutter. If I just simply want to cut the DNA and splice it so that I can see maybe the size of the sample or if it has those sites, then I can use something like SMA1, which creates blunt ends. There are no sticky ends here. So this is not a good enzyme to create recombinant DNA samples where you're taking DNA from one place and then splicing it into another vector. This would not work. So DNA fragments with compatible sticky ends can be ligated together. And when that happens, we use an enzyme called ligase, and we have to add ATP to be able to combine them and put them back together again. So the sticky ends need to match, so they have to have the same complementary sticky ends to be able to do that in the research lab. Okay. DNA fragments with blunt ends can be ligated together. It's just a little different. You're ligating at a different point. And like I said, this allows ligation to occur in different locations than normally present. So you can ligate these types of, this example or this something like this. So sequence recognition and cleavage. Sometimes there are enzymes that are not 100%, don't have to have 100% match for the bases that are in its recognition site. So here we have an example of AVA2 and AVA1. And what does this mean? It means basically that here where there's a W, it can basically have any base in that section as long as it has 2 G's and 2 C's with a gap, a 1 base gap in the middle. So it doesn't matter what base is here, it needs to bind to these two to be able to cleave it. For AVA1, you need to have specific pyrimidines, so either T's or C's, and a specific purine, A or G, in these spots to be able to cleave the sequence. So you basically need, you can have all these different combinations. Okay, so these are enzymes that are a little bit less strict with what base it needs to have. So the recognition site for SFI1, if you notice, SFI1A has a different, the beginning and the ends are the same for the SFI sites, but it's this center region that's different. So SFI in general can have any bases in this region, and for our purposes, SFI-A has this sequence and SFI-B has this one. And again, I told you that you could buy these recognition sequence, these enzymes, and this is just a page from one of the distributors. Okay, so we describe the activity of a restriction enzyme based on the units of restriction enzyme that actually digest within a certain period of time. So NEB, New England Biolabs, defines a unit as 1 unit of restriction endonuclease activity is defined as the amount of enzyme required to completely digest 1 microgram of substrate DNA in a total reaction volume of 0.05 milliliters in 1 hour with using the buffer provided. Now, it specifies that it uses the buffer provided because all of these enzymes have specific pH and buffer requirements. So you must be using the correct buffer to get optimal activity. So here's an example of optimal conditions for an enzyme. This is just one example. Like I said, there's many others. Some of them require different ions to be present, like magnesium, or reducing agents and temperatures that they work at. And they're high and low temperatures, which means that they can work, you know, their optimal temperature might be 37 degrees, but it can function up to a certain temperature. So in this case, before we're setting up a restriction enzyme, we need to know that we're using the right buffer solutions and that they match the restriction enzyme you're working with. Improper conditions can cause denaturing of the enzyme, or another something else that could happen is star activity, which is non-specific digestion. When you don't have the right conditions, or you leave an enzyme digesting DNA maybe overnight or too long, sometimes the enzyme just starts to randomly digest in different places. And that can cause a lot of confusion when looking at gels.